Bismillahirrahmanirrahim. Assalamu alaikum. I am Dr. Ayesha Jamal, lecturer of Botany from Government Degree Girls College, Block 12, Gulistan-e-Jauhar, Karachi. The topic of my lecture is Chromosomes and DNA from Biology, book chapter number 6 in HEC part 2. Students, chapter 6 is very important from examination point of view as major portion of botany paper includes questions from genetics. Either these are MCQs, short questions or long questions. If you prepare the genetic portion well, you can cover major portions of biology paper without any problem. So let's start with our lecture. Let me first give you a brief introduction of the topic. As you have studied earlier, about the cell theory, which comprises of two main parts. First, cell is the fundamental unit of life, and second, cells arise from pre-existing cells. It means for producing life, previous or old cells have to divide. This process is known as reproduction. Reproduction allows the continuity of life. Without reproduction, life would come to an end. In chapter number 4, you studied the process of reproduction in detail. Continuity of life forms on Earth is based on an organism's success in passing genes on to the next generation. All life forms share at least the same two structures, that is nucleic acid and proteins. All organisms have reliable means of passing genetic information to offsprings through reproduction. Offsprings receive the same genetic information as parent, but some variability do exist. Actually, the entire genetic information is contained by the chromosomes which store this information in the blueprint of life DNA. DNA not only develops and maintains an organism but also transfers its command onto the next generation. So the main objectives of this lectures is to make you understand the basic concepts about the continuity of life. Throughout the lecture, we will focus on the following points, that is, what are chromosomes and DNA, where are chromosomes located, structure of chromosomes and DNA will be discussed. Along with the structure, we will also discuss the function of chromosomes and DNA. First, we will discuss about chromosomes. The term chromosomes is a combination of two words, that is, chrome means colored and soma means body. Actually, in 1882, a German embryologist, Walter Fleming, was examining the rapidly dividing cells of salamander larvae that was treated with Parkin's aniline dye. During this examination, he observed certain thread-like structures appeared much darker than the rest of the organelles. So we can say that chromosomes were discovered by Walter Fleming, but the name was given by Wilhelm Welder. According to him, these are not colored themselves, but they easily accept dye or stain and take patterns of light and dark when exposed to different stains that are used to identify chromosomes. These chromosomes are macroscopic thread-like structures in the cell and they carry hereditary information in the form of genes. The chromosomes are located inside the nucleus of every eukaryotic cell. In case of prokaryotic cells, these are present as circular genetic material in the cytoplasm because prokaryotes do not have a well-defined nucleus. This picture shows the natural scheme of the whole genetic material inside a eukaryotic cell. The chromosomes cannot be observed unless and until the cell divides. In interphase mitotic division, chromosomes are very thin structures and termed as chromatin. As cell division proceeds, they appear as thick thread-like structures due to condensation. And the chromatin network appears in the form of individual chromosomes. First of all, we will discuss the morphology of chromosomes, means how does a chromosome appear and what are the different parts of each chromosome. Each chromosome consists of two thread-like structures known as chromatids. This is one chromatid and this one is the second chromatid. These two chromatids are separated from each other along their whole length, except at a single point. This point is called centromere. You can say centromere is a point of attachment for two chromatids. Chromosomes are present in pair form. Chromatids are the part of one chromosome. It means pair of chromosomes will have four chromatids. Two chromatids of one single chromosomes are called sister chromatids. But the chromatids of two homologous chromosomes are called non-sister chromatids. Now in this picture you can see the ends of a chromosome which appears darker in color. These ends are known as telomeres. Telomeres are the sections of DNA found at the ends of each chromosome. Telomeres serves three major purposes. They help to organize each chromosome in the nucleus. They protect the ends of the chromosomes. If telomeres were not there, the chromosomes may end up sticking to other chromosomes. They allow the proper replication of chromosomes during cell division. Actually, we can classify chromosomes on different bases, like on the basis of centromere position or on functional basis. As I told you, that centromere is a region on the chromosomes where the spindle fibers bind during the process of cell cycle. Depending on the position of centromere, chromosomes can be classified into four basic types. These are the metacentric, submetacentric, acrocentric, and telocentric. In case of metacentric, the centromere is in the center and it divides the chromosome into two equal arms. In submetacentric condition, the centromere is present slightly away from the center or nearer to one end of the chromosome. As a result, chromosome is divided into one shorter and one longer arm. In acrocentric chromosome, the centromere is present very close to one end of the chromosome, thus it forms one extremely short and one very long arm. And last one is telocentric. In telocentric chromosome, the centromere is present at the terminal end of the chromosome. This chromosome appears to have a single arm. In the last slide, I used the term homologous chromosomes. Actually, it is a pair of chromosomes that contain the same gene sequence. The two chromosomes in a homologous pair are very similar to each other. They have the same size, shape, and centromere position. One chromosome in a pair is inherited from organism's father, and one chromosome is inherited from organism's mother during fertilization. Afterwards, each chromosome produces its sister chromatid by replication. Here it should be noted that homologous pair have the same sequence of genes. As you can see here, same sequence of genes for each character, but alleles may be different. After replication, the newly formed sister chromatid of each chromosome will have the same allele for each gene, as you can see over here. Understood? We can also differentiate chromosomes into two categories on the basis of their function, that is autosomes and sex chromosomes. Autosomes are the pair of chromosomes that regulate the somatic characteristics of the body. And sex chromosomes, also known as allosomes, they are the pair of chromosomes that determines the sex of an organism, as they regulate the sex-linked traits. For example, have a look on the karyotype of a human cell. There is a total of 46 chromosomes, or you can say 23 pairs in the human genome. Out of all these 23 pairs, 22 pairs from here to 22, these 22 pairs of chromosomes are all autosomes, but the last 23rd pair is known as the sex chromosome. The, these sex chromosomes determine the sex and sex-related characteristics of an organism. This is the basic difference between autosomes and sex chromosomes. Sex chromosomes are designated as X and Y. If the if both the chromosomes in a pair are X and X, then the individual will be female. But if one chromosome in the pair is X and other chromosome is Y, then the individual will be male. And the Y chromosome is smaller in size. Okay. Now we have discussed the general appearance of chromosomes. Now come to its chemical composition. Eukaryotic chromosomes are composed of chromatin, which is a complex of DNA, proteins, and RNA. Major portion comprises of DNA and proteins, while a small amount of RNA is also present because they synthesize proteins. DNA is actually a double-stranded polynucleotide chain. What is polynucleotide? Poly means many and nucleotide is the basic unit of DNA. There are so many nucleotides, billions of nucleotides combined together and they form the strands of DNA. Second portion are the proteins. Now each nucleotide is basically made up of three different molecules that is the phosphoric acid, deoxyribose sugar, and a nitrogenous base. In every nucleotide, phosphoric acid and deoxyribose sugar is same, but nitrogenous base may differ because there are basically four types of nitrogenous bases, that is adenine, guanine, cytosine, and thymine. Each nucleotide contains any one particular type of nitrogenous base at a time. The whole complex chemical material is present inside the nucleus of the cell, which cannot be seen through our naked eyes. So to better understand the ultrastructure of chromosomes, the DNA must be unwound. हमें DNA को खोलना पड़ेगा, हमें chromosomes को खोलना पड़ेगा. To know the structure of eukaryotic chromosomes, metaphase chromosomes are preferred. They are easily observed under microscope after staining. Just have a look on this picture. On the left side, there is a chromosome. This enlarged highlighted portion appears as thread-like structure. If this portion is unwound, अगर हम इस portion को खोलते चले जाएं, then each component all the components are gradually appearing more clearly. आहिस्ता आहिस्ता में हमें छोटे-छोटे components इसके clear होते हुए नजर आएंगे. Maximum unwinding reveals that there are numerous repeating units which make the chromosome. बिल्कुल बहुत जब पूरा के पूरा हम इसे खोल लेंगे, मैक्सिमम अनवाइंड कर लेंगे, तो हमें पता चलेगा कि बहुत छोटे छोटे से यूनिट्स हैं जो मिलके पूरा क्रोमोसोम बना रहे हैं. Every component is arranged in an organized manner. एक पूरा एक ऑर्गेनाइजेशन है. DNA है, DNA is wrapping or it wraps around proteins. Proteins form beaded structures. These beaded structure coils to form chromatin. Chromatin makes loops and again coil together forming supercoils. These supercoils bunch together to form the whole chromosome. So every level of packaging is well organized. Here we will discuss each level separately. The DNA in the chromosome is present in the form of two long strands. If these strands are stretched, It would be approximately 2 meters long. This entire length cannot be fit into a small cell. So this DNA is coiled again and again to fit itself in a nucleus. When the DNA is stretched, it resembles a string of beads. यानी अगर हम इस strands को खोलें तो हमें एक beaded structure नजर आएगा. अब ये beaded structure जो है वो किस चीज़ से बने हैं? अगर हम इसको अनकॉइल करते हैं, खोलते हैं, तो हमें ये beaded structures clearly नजर आ जाएंगे. अगर हम एक beaded structure को स्टडी करें, देखें, तो हमें पता चलेगा कि there are special proteins which are known as histone proteins present. 8 molecules of histone proteins are present in the center around which a portion of DNA duplex is coiled. यानी DNA अपने आप को 8 molecules of histones के सराउंडिंग में अपने आप को रैप करता है. इस रैपिंग की वजह से, इस कॉइलिंग की वजह से एक पर्टिकुलर स्ट्रक्चर, बीडेड स्ट्रक्चर बनता है. इस बीडेड स्ट्रक्चर को हम न्यूक्लियोसोम कहते हैं. न्यूक्लियोसोम में क्या क्या चीजें आ जाएंगी? सेंटर में एक 8 मॉलिक्यूल्स ऑफ हिस्टोन विच इज ऑल्सो नोन एज ऑक्टेमर, यानी सेंटर में ऑक्टेमर होगा और उसके सराउंडिंग में डीएनए रैप हुआ होगा. अच्छा, इस बाइंडिंग के लिए ज़रूरी है कि एक पॉजिटिव चार्ज हो और एक नेगेटिव चार्ज हो. तो होता यह है कि द हिस्टोन प्रोटीन्स आर मेड अप ऑफ अमाइनो एसिड्स दैट आर arginine and lysine, and these two, arginine and lysine, are basic in nature, so that's why they have positive charge on them. It means histones are positively charged. और ये जो डीएनए डबल हेलिक्स स्ट्रक्चर है, जो डीएनए स्ट्रैंड्स हैं, these are negatively charged due to the presence of phosphate group. ये हम आगे चल के डिस्कस करेंगे इसका स्ट्रक्चर. So the DNA duplex is strongly attracted to the histones and wraps tightly around the histone core of each nucleosome. तो न्यूक्लियोसोम के लिए बनने के लिए एक strong attachment है between DNA and the histone proteins. Now this is one structure of nucleosome, this is another nucleosome and this is another nucleosome. यानी एक न्यूक्लियोसोम बना, उसके बाद डीएनए थोड़ा सा आगे बढ़ेगा और फिर एक और न्यूक्लियोसोम बनाएगा. The small portion of DNA between two nucleosomes is known as linker DNA. यानी it means कि दो न्यूक्लियोसोम को जो डीएनए का पोर्शन लिंक कर रहा है उसको हम linker DNA कहेंगे. And it comprises of 200 nucleotides. It means after every 200 nucleotides the DNA duplex wrap itself around octamer. अपने आप को दोबारा से हर 200 न्यूक्लियोटाइड्स के बाद जो डीएनए है वो अपने आप को रैप करेगा. ओके, नाउ अब इस तरीके से बहुत सारे न्यूक्लियोसोम्स बन गए, लेकिन ये न्यूक्लियोसोम्स इसी तरीके से दूर दूर नहीं रहेंगे, बल्कि ये आगे चलकर अपने आप को मजीद कॉइल करेंगे. यानी ये अभी हमें दूर दूर नजर आ रहे हैं, लेकिन जब ये आगे चलेंगे तो ये और थोड़े से क्या करेंगे, दोबारा से कॉइल होंगे, मजीद ये अपने आप को कॉइल करेंगे. यहाँ पर जो विड्थ है इट इज 2 नैनोमीटर. स्ट्रैंड्स की जो विड्थ है वो 2 नैनोमीटर है, लेकिन जैसे जैसे कॉइलिंग होती जाएगी आगे आगे ये विड्थ बढ़ती जाएगी इस स्ट्रक्चर की. यहाँ पे 2 नैनोमीटर था, लेकिन आगे चलकर जब न्यूक्लियोसोम्स बने और न्यूक्लियोसोम्स जब अपने आप को कॉइल करेंगे तो दे विल स्टार्ट फॉर्मिंग क्रोमैटिन. अब क्रोमैटिन बनाना स्टार्ट कर देंगे. नाउ दे विल रैप न्यूक्लियोसोम्स बैक इनटू अ कॉइल. अब ये न्यूक्लियोसोम्स कॉइल करेंगे, पैक करेंगे अपने आप को कॉइल्स में, दैट ट्विस्ट इनटू अनदर लार्जर कॉइल. यानी पहले छोटे कॉइल्स बनेंगे और उसके बाद फिर बड़े कॉइल्स बनेंगे, एंड सो ऑन, एंड प्रोड्यूस कंडेंस्ड सुपर कॉइल्ड क्रोमैटिन फाइबर्स. यानी कॉइलिंग बढ़ते बढ़ते, सुपर कॉइलिंग होते होते इस तरह फाइबर लाइक स्ट्रक्चर्स बन जाएंगे और क्रोमैटिन फाइबर के तौर पे हमें नजर आएंगे. जैसे जैसे मैंने बताया पहले विड्थ कम है, लेकिन जैसे जैसे हम आगे बढ़ते जाएंगे, कंडेंसेशन जैसे जैसे ज़्यादा होती जाएगी, वैसे वैसे इसकी क्रोमैटिन नेटवर्क की विड्थ बढ़ती जाएगी। यहाँ पर 2 नैनोमीटर था, आगे चलकर 30 नैनोमीटर पर ये विड्थ आ गई। उसके बाद मज़ीद जो है जब सुपर कॉइल्स होंगे और जब ये लूप्स बनाने स्टार्ट करेंगे, then the the width of this super coil will become 300 nanometer। ये 300 नैनोमीटर पे चला जाएगा। Now these 300 nanometer fibers are compressed। अब पहले तो ये coiling और super coiling हो रही थी, अब यही coils अपने आप को करीब करेंगे और ये compressed होंगे and they are again folded, और दोबारा से अपने आप को fold करेंगे to produce wide fiber, यानी उससे और मजीद चौड़ा और विड्थ में ज़्यादा जो इसकी विड्थ है वो बढ़ती चली जाएगी, यानी 300 नैनोमीटर विल बिकम 700 नैनोमीटर। अब ये 300 नैनोमीटर का स्ट्रक्चर मजीद सुपर कॉइल होकर ये 700 नैनोमीटर स्ट्रक्चर बना रहा है। एंड देन the tight coiling of the fibers produces finally the tight coiling of the fibers produces the chromatid of the chromosome। और finally ये tight जब coils बहुत एक दूसरे के करीब आ जाएंगे और tight हो जाएंगे, तब ये क्रोमैटिड बना लेंगे, एक क्रोमोसोम का क्रोमैटिड बन जाएगा। एंड द लास्ट विड्थ ऑफ द क्रोमोसोम दैट इज द 1400 नैनोमीटर। ये अब ये 1400 नैनोमीटर पर आ गई, यानी जो मिनिमम विड्थ है स्ट्रैंड की वो 2 नैनोमीटर है, which is the width of DNA strand। लेकिन आगे जब इसकी कॉइलिंग और सुपर कॉइलिंग और जो कंडेंसेशन हो जाएगी और डुप्लीकेटेड फॉर्म में आ जाएगा, देन द विड्थ विल बिकम 1400 नैनोमीटर, which is the final structure of the chromosome। Due to this packaging, two distinct portions of chromosomes are seen when observed under microscope: light condensed regions and the dark condensed regions. The light condensed regions of chromosomes are known as euchromatin. This is the structure of the euchromatin. This is the area of the euchromatin. And the tightly compact regions of chromosomes, which are known as heterochromatin. Euchromatin, this portion is condensed only at the time of cell division. At all other times, the euchromatin is an open configuration and its genes can be activated. Whereas heterochromatin, they are transcriptionally inactive in nature and sometimes they remain condensed permanently so that their DNA is never expressed. Euchromatin are the regions where 30 nanometer fiber forms radial loop domains in the chromatin network, whereas heterochromatin are the portions where the radial loop domains is compacted even further, so making it more condensed area. Now come to the structure of DNA, which actually makes the whole chromosome. The three-dimensional double helix structure of DNA was suggested by James Watson and Francis Crick in 1953. This double helical structure consists of two strands twisted together, and each strand is a long sequence of nucleotide. As I told you before that DNA complex or DNA strand is a polynucleotide chain. Each nucleotide consists of 3 molecules: a phosphate group, a deoxyribose sugar molecule, and a nitrogenous base. Now the phosphate group and sugar molecule in each nucleotide remains the same, but each nucleotide may contain different nitrogenous base. As I told you that nitrogenous bases are of 4 different types. Nitrogenous bases are of 4 different types, that are cytosine, thymine, adenine, and guanine. Now cytosine and thymine are known as the pyrimidine bases, whereas adenine and guanine are termed as the purine bases. you can see the difference in their structures. These pyrimidine bases that is cytosine and thymine, they are single ring structure, whereas purine bases, adenine and guanine, they are having double ring structure. In each nucleotide, the phosphate group is attached to the fifth carbon of the sugar molecule, and the nitrogenous base is attached with the first carbon of sugar molecule, and this makes up the whole unit. This one unit is the base of the whole DNA structure. DNA molecule is made up of two long chains of polynucleotide. Each of these chains is known as DNA chain or DNA strand. In a chain, a phosphate group attached covalently with the sugar molecule of the next nucleotide, forming a long polymer of nucleotide, means nucleotides are attached end to end and they form the long chains. In this way, they make up the backbone of the DNA. You can say the backbone of DNA is based on a repeated pattern of sugar group and a phosphate group. The three-dimensional structure of the DNA arises from the structure feature of its polynucleotide chain. These two chains are held together by hydrogen bonding between their bases. All the bases are on the inside of the double helix and the sugar phosphate backbones are on the outside. Here it should be noted that there is a specific complementary base pairing between the chains. Two-ringed purine base is paired with a single ring pyrimidine base. Like adenine always pairs with thymine and guanine always pairs with cytosine. There are two hydrogen bonds between adenine and thymine, whereas guanine and cytosine are held together by three hydrogen bonds. Because each base pair is of similar width, the two polynucleotides are at an equal distance of 2 nanometer away from each other along the whole DNA molecule. Each strand has one end at which there is a phosphate group, which is attached to the fifth carbon of ribose sugar. This end is marked as 5' end. On the other end of the strand, there is a free hydroxyl group attached to the third carbon of a ribose sugar. This end is marked as 3' end. Double helix. In this double helix, the direction of the nucleotides in one strand is opposite to the direction in the other strand. So we can say the DNA double helix is anti-parallel, means that the fifth end of one strand is paired with third end of its complementary strand and vice versa. This determines the polarity of the molecule and helps in replication of DNA and gene expression. We can say that DNA is the master plan for life on Earth and the source of the wonderful variety we see around us. It is a complex molecule that contains all the information necessary to build and maintain an organism. It serves as the primary unit of heredity in organisms of all types. During reproduction of organisms, a portion of their DNA is passed along to their offspring. This transmission of DNA actually ensures a certain level of continuity from one generation to the next. But it still allows slight changes that contribute to the diversity of life. Chemically, your genetic endowment is the DNA you inherited from your parents. DNA molecule contains each person's unique genetic code. It holds the instructions for building the proteins that are essential for our bodies to function. Students, I have tried my best to cover the important concepts of DNA and chromosomes in this lecture. Hope you understand each and every part. Thank you and best of luck.